some like if you miss or you want to recall the sessions, you can directly take it from there. Yeah. So yeah, welcome to the week 2 of this problem solving session. And so this time what we are looking is more on PCR, yeah, polymerase chain reaction. So the first one was PCR fundamentals, then something about the real-time PCR, so 2 videos on it, and then predominantly the variations of PCR, keeping in mind the different applications and its properties. Yeah. So, and also we learned about some how to quantify and how to analyze PCR data and what are the parameters involved in it. Yeah. So we'll just recall some of it, and then we will directly go and discuss through the problems that we have prepared. Yeah. So coming to the uh, what we learned as a core principle is it has it's the mechanism draws its mechanism is based on exponential DNA amplification. So it's kind of a in vitro mode of DNA replication that we see, yeah, in our body. It's just what we do it outside the body. So it's an exponential DNA amplification through thermal cycling. Thermal cycling because it has a lot of um, cycling of the temperature involved in it. So it's a three-phase process: denaturation, annealing, and extension. And some of the critical components that are involved as polymerase, you have DNTPs, primers, and then you have magnesium buffer also. So now they have made a lot of innovations in this, and you have different kinds of PCR, the hot start PCR, touchdown PCR, and some other methodologies like droplet PCR also that you can see. Yeah. Coming to the quantitative techniques that drawn a lot of attention during this COVID time, you have rebuilt real-time PCR, yeah, that we saw, and in that you can actually quantify the viral load using the CT number that people used to tell. So how this real-time PCR works is more on on the labels, yeah, the fluorescent probes, which are basically the labels. So this fluorescent probes, usually cyber green, TACMAN, and a lot that is discussed in the videos. This enable real-time monitoring. So with the PCR machine, like thermal cycler unit, if on top of it, if you add a detection platform in which you can actually excite and collect the fluorescence using the different photomultiplier tubes or avalanche detectors. If you can detect those fluorescence signals, what we get is a real-time PCR. So it's thermal cycler plus the detection unit. What we end up with a unit called as real-time PCR. Yeah? So in which these fluorescent probes are used. So basically these fluorescent probes are in a way the label. You actually can't you are actually not quantifying the um, DNA or nucleic acid material that is getting amplified, but actually you are seeing the fluorescent probes, fluorescent label. So those acts as a label. So you are indirectly detecting it. Yeah. So you will need a calibration curve also to see how much fluorescence is getting um, fluorescence is getting like getting produced corresponding to what copy number of the nucleic acid copy number. Yeah. So you have CT value quantification with standard curves usually, and then you have absolute versus relative quantification methods also. And then you have digital PCR, which is coming up a lot in the current time. So you have you basically will partition or divide your sample in droplets, and then will do the quantification, and actually you can count the number of droplets, yeah, and giving positive signal and such. So this essentially works with some Poisson distribution, and you can correlate it with the final concentration that you have of the sample. So in this you have enhanced sensitivity for rare alleles and copy number variations. Yeah. coming to some specialized amplification methods, you saw the reverse transcription PCR, in which you can actually convert the RNA from predominantly viral samples usually, where you convert it to complementary DNA and then you do the PCR. So there are two approaches, yeah, one step versus two step, and the advantages of two step has been given and why it is used mostly. Then you have long range PCR, in which you can actually amplify oligos or DNA which is more than 5 kilobase per. So in which you need high fidelity polymerases. So these are used for usually for sequencing applications and such, or mutagenesis, site directed mutagenesis applications. And if we talk about some application based, in which you have allele specific PCR, in which you can actually determine the single nucleotide polymorphism detection through the primer mismatch, or the ARMS PCR or multiplex PCR, in which you can have multiplex detection of like simultaneous detection of multiple pathogens. But usually multiplex PCR has not seen a lot of light because it has its predominant challenges in terms of it. It has there are a lot of non-specific effects that can generate using this multiplex, and also you have to take care of the annealing and all the temperatures that are involved in it. So all the primers should work in the similar temperature range. So that's a it's a like a design problem that this multiplex PCR has. So yeah, then you have some molecular techniques using like colony PCR for bacterial and inverse PCR. So inverse PCR you can actually amplify and generate flanking unknown sequences. Yeah, so it is needed in some in a lot of applications here. Then you have some in situ PCR, in which you can cellular localization. You can basically take up cellular localization of nucleic acids and also from tissues, yeah, tissue architecture during amplification. And an interesting amplification method is also ligase chain reaction, in which you use ligase plus polymerase. So this ligase is something which is very sensitive to the mutation. So if two strands are coming together, okay, and so for example you have this sequence here, and you have two primers coming together, okay. So uh, this ligase should act here, yeah. So what it will do is, if this bases are so this is one strand, this is two strand, and this is so this should match exactly. If there are some gaps or it is mismatches, this ligase won't work. So this efficiency of this ligase is very is dependent on the mutations. Or so basically if you are using these as primers and this as a target, if it exactly matches, if this primers exactly matches with the target, then only the ligase will work, and then you can actually amplify it. So it's kind of a two step kind of what do you say, a two step authentication, kind of a two step lock, in which you have to tick both of this system, and then only it will go ahead and amplify. So yeah, that's what ligase chain reaction is. And you also saw the melt curve analysis, different kinetics, yeah, kinetics, how this curve happens, and then cell gel electrophoresis depending on the molecular weight, yeah, in which you run a ladder, yeah, and then corresponding to it you see your products. Some emerging directions are microfluidic. So now people are putting PCR on a chip. So PCR plus microfluidics. You have CRISPR integrated cell detection systems. People are doing also single cell PCR, and then you can also learn about some isothermal application, like You must have heard about LAMP, one very famous isothermal amplification. LAMP, or you can also search about rolling circle amplification. So these are something that are currently getting a lot of attention. So isothermal means isothermal means same temperature, same thermal means temperature, or preferably preferably room temperature, which this same temperature people are currently working on using it for room temperature, so that you can actually use these methods in resource limited settings, yeah, because PCR involves this thermal cycler and detection methods, and which maybe in some parts of some resource limited settings it can be bit difficult to operate, yeah. So people are also working on isothermal amplification techniques, and actually you have a term on it is called as NAT, yeah, nucleic acid amplification tests. So it's NATs. So all this comes under this umbrella of NATs, nucleic acid amplification tests. Okay. So I think that's all for summary. We can actually directly go to the problems. The final concentration of Taq polymerase is to be 0.01 units per microliter in a 50 microliter PCR. If the enzyme is supplied as 5 units per microliter, how much enzyme would you add to the reaction? So the way is you solve it and you put the answers in the chat box. After 5 minutes, we will discuss the answer, and then we can discuss, yeah, how people have solved it.
இன்னும் ஒரு பாக்கெட் இருக்கா 4 வாங்கி கீழே அந்த சப்பாத்தி டப்பா எடுத்து பாருங்க. அதுல சப்பாத்தி டப்பா எடுத்து வெளியே பாக்கெட்டு
Others, are you solving it? Okay, how you are solving it? Anyone? Okay, so yeah, first we will take c1 equal to, so let's first write it down what we know. Yeah, so initial concentration is 5 units per microliter, yeah, and you need 0.01 units per microliter, and V final is 50 microliter. So Ci into Vi equal to Cf into Vf. Ci is 5 into Vi. We have to find out Cf into 50 microliter. So Vi is 0.1 microliter. Yeah, well and good. Okay, we'll go to the next one then. Primer dimers result from, okay, so can anybody tell the answer and explain why it is? Anyone? You can unmute and tell. So, so the answer is D. So what happens is when you have 3' complementarity, what happens is this primers anneal to each other. Yeah, they anneal to each other. Yeah, and they form stable structures. And in sometimes they can also form short amplification products also, yeah. So as a result, what you this if it starts to form short amplification products, you essentially are competing with the amplification of the template sequence, yeah, or the target sequence. So we should avoid the 3' complementarity, or basically the primer dimer condition, yeah, it's true. Okay, should we go ahead? Okay. Which control is run to detect contamination? Okay. Okay. What is okay? Can anyone define? Anyone define what are these the terms? Yeah. Negative control, positive control, molecular weight marker, and reagent link. Anyone? So controls are like important part. So it's always that we focus on the test, but controls are something that validate the test. So designing a control is very important. Anyone? What is negative control, positive control? Yeah. So the answer is reagent blank. So negative control, okay, negative control, it contains all the reaction. It 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 contains all the reaction components. Yeah, and then you have the only without the target. Yeah, without the target. So and then you have positive control. So positive control is used to verify that the reaction works properly. So positive control is something that you want to check that it should come positive for the system. Yeah, so you can say it is for checking that the reaction works, and it is not for contamination detection. So and negative control, it should not have the target, and it is so it's different than detecting the contamination. And then you have the molecular weight marker. So it's molecular weight marker is for determining the size of the DNA fragments. Right, just someone is, yeah. So and then you have reagent blank. So reagent blank is a mixture in which it contains all the solvents and reagents which is involved in the analytical procedure, but it will not contain any analyte or sample material. So it represents the background only from the reagents and the sample alone, or the analytical system alone. So basically, if there is any contamination or DNA in the reagent, or the in the polymerase, or in whatever buffers that you are involved, if it is getting contaminated, or if it is getting showed up in that, you can actually only check through the reagent blank. So the purity or any background interference you want to check, you have to go for the reagent blank run. Yeah. Okay. Non-specific extra PCR products can result from.
Hello.
Yeah.
Yeah, so the previous question, the negative control, if it is positive, even that indicates contamination.
Yeah, but then you have to go for the reagent blank. Then you can see whether so you can see whether there is something on the only on the reagents. Yeah, something that is coming, because then there can be some primer based effects also that can come with the negative control. So to check that, you go for the reagent blank. You won't in that you won't have the primers also, only the reagents. Is it okay?
Okay.
Yeah. So non-specific extra PCR products can result from. Yeah, the answer is mispriming. Can anyone tell why it is the correct and others are wrong? So what happens in mispriming, and you have to tell why other options are incorrect. That's all. Anyone? So what
So the
happens?
primer is
Hmm.
binding to a non-target area of the DNA
True.
because of
So
its low specificity.
okay, so that happens in mispriming, yeah. So you are getting incorrect binding sites, yeah. So and unwanted amplification. So that's what happens in mispriming. And what happens if you have high annealing temperatures? Does it help, or does it uh, like is it does it contribute to non-specificity?
A primer will not be able to bind.
Yeah, but if you have, so it's like okay, yeah, true. But if you have actually like I should have done like kind of high means, yeah, I should have maybe I could have mentioned, but high means if you have actually have higher or kind of high annealing temperatures, you usually have it usually will reduce non-specific products. So when you go for higher annealing temperature, I'm not telling too high such that it doesn't it touches the denaturing temperature, not that. But when you have annealing temperature, you have here it is mentioned, yeah, annealing temperature, when it's the temperature is suitable for annealing, but if you are keeping it high, what happens is you essentially will get non-specific products. You essentially will get specific products. Sorry. So yeah, mispriming is correct. This actually will help reduce. This will reduce non-specificity. And high agarose anyway, it's for separating. Yeah, it's for separation of. Just wait, somebody has. Yeah. So it's for separating DNA fragments. And what happens if we omit MgCl2?
The activity of the tag polymerase will be reduced.
True. So yeah, so magnesium is needed for activity of DNA polymerase. So you essentially you will have inhibition, PCR inhibition, yeah, due to the DNA polymerase activity. So magnesium is essential for PCR DNA polymerase activity. So the answer is mispriming. Okay. With using which of the following is an appropriate way to avoid PCR contamination? Okay, anyone want to answer? So the answer is the answer is C, a separate area for PCR reaction setup. Yeah. So having a dedicated PCR setup is the most appropriate way to avoid PCR contamination. But yeah, why are other options wrong? Yeah, that's what have to be also seen. So having a dedicated, so what it gives is physical separation. Yeah, so it has its own physical separation benefits. So you won't have any cross contamination between pre and post PCR samples. You have a clean environment for reaction preparation, and you essentially don't have any risk of this amplicon getting carry over in other reactions. What happens if on high fidelity? So high fidelity polymerases usually have very high accuracy on DNA replication. It has very so it improves DNA replication. Yeah, less mutation also, but it doesn't address the contamination issue. Yeah. Then you have hot stack PCR, in which you can reduce non-specific because of low that temperature barrier, no non-specific amplification. But same thing, it doesn't address the contamination thing. When you have fewer PCR cycles, you have reduced yield. Yeah. So as a result, have less sensitivity also. And it doesn't address the contamination. So the appropriate answer is C. Is it okay? So how many copies of target are made after 30 cycles of PCR? Okay. So maybe I should the better better question it's if it has initial it is one, yeah, initial one copy is only there. So So usually the answer is 2 to the power 30. Yeah. So if it is one copy, assuming it is one copy initially. So usually the formula is n into to the power n. So n means number of copies initially. Yeah. Initially. And then you can multiply it into the power n, where n is number of cycles. So this 2n are different here. Which of the following is a method for purifying a PCR product? Yes, raised your hand here. You have any doubts? You want to ask something? Okay. The answer is putting the reaction mix through a spin column. Yeah. Can anybody tell the answer? Why it is? You people have answered also, some of you.
It is because silica specifically binds to the PCR product, the DNA. That's why we use spin column, which is coated with silica membrane.
Yeah. So yeah, it kind of concentrates the DNA, and you can actually recover a pure product out of it. So the answer is spin column. Others are not relevant essentially. Yeah. DPC is something do you know what is DPC, you people? You may see that the some so when doing with when you're working with DNA you or nucleic acid as such, you will have something called as nucleus free water. Yeah. So in this nucleus free water there will be it will be written it is DPC DPC treated. I don't know, I don't recall the full form of it, but it will be written DPC treated. Yeah. So essentially this DPC treatment is done such that it makes the water ideal for RNA work. So RNA basically it will inactivate any RNAs or such there. So there in that or any nucleases maybe as such. But yeah, you people can check it. But yeah, mostly that nucleus free water will have this DPC treatment which is done. Okay, should we go ahead? Yeah, okay. Diethyl pyrocarbonate. Okay, thanks. Yeah. So in contrast to standard PCR, real-time PCR is diethyl pyrocarbonate. Okay, thanks. Yeah. So as we discussed, it is quantitative. Uh, yeah, the difference is something that is quite evident that standard PCR essentially only will have a thermal cycler unit in which you can change the temperature. In a real-time PCR, you will have a detection unit attached to it in which you can excite the fluorescence probes and also view them or quantify them using a detection unit. So that makes it real-time PCR. So in real-time PCR, fluorescence is not generated by which of the following? Yeah, the answer is D. So FRET, what it is? It generates fluorescence through this fluorescence resonance energy. Yeah. So yeah, that generates using this receiver donor concept when you have them very closely. This closely spaced, and then you have TACMAN, in which it has 5 prime nucleus activity, yeah, which uses 5 prime nucleus activity and generates fluorescence. And then you have cybergreen, which I believe intercalates, intercalates and generates fluorescence. Yeah. It is green because why it is called cyber green? Does anyone know why it is cyber green? Yeah, you want to answer? No? Yes, tell them why it is cyber green. True. The emission wavelength is green, yeah. So it gives green light. Then what it absorbs? What what light then it gets excited?
Blue light.
Huh? Someone someone told anything? I couldn't hear. Yeah, it's blue light. Yeah. So it absorbs blue light and emits. So it gets excited at on the blue light, and you can actually get green light getting emitted. So the exact, I think, exact wavelengths I'm not sure, but maybe it will be around, it will get excited around 495 to 500 nanometer light, which is blue, and maybe will show emission around 520 to 530. So that's how these filters, what this real-time PCR machines will have, are those filters, yeah. And essentially you can have multiple dyes, and then you can actually use these fluorescence filters to check or quantify. And this similar principle is there with the fluorescence microscope also. The full form of cyber, yeah. Actually, I don't know. I know that it's kind of cyanide dye, yeah. But full form, I really don't know. Maybe if any one of you can check, yeah, then you can write it in the chat box. I'm not aware actually. Okay, so answer is yeah. Yeah. So it's yeah. So it's used to quantify double stranded DNA, and yeah, you can use it in flow cytometry. True. So the answer is here is TTH polymerase. Yeah. So yeah, which of the following factors affect the fidelity of DNA polymerase in PCR? So this I believe is a question from GATE. Yeah, I think I took it from the GATE question paper previous year. Any of the previous years, yeah. I don't recall. Okay. So the answer is P and Q. Yeah. A. Anyone would like to answer or discuss?
Due to pH also there are chances of denaturation.
Yeah. pH. That's what pH. P and Q. Yeah. That's what I'm not.
The
P and Q.
concentration of Mg2 plus and pH, which decides the enzyme activity, it will
Activity.
reduce if it is altered, right?
Yeah. That's
That's
the answer. That's the answer. Yeah. That's right. P and Q. Yeah. It's not the annealing temperature, because annealing temperature is something that controls the primer binding. So it doesn't directly influence the polymerase activity. So if it can only affect the primer binding. Yeah, specifically, like that's what annealing temperature is. Annealing temperature only will have effects on primer binding specificity. Otherwise, Mg2 plus and pH is something that is that acts on, that directly affects the activity or fidelity of DNA polymerase. Yeah. True. So the answer is P and Q. Is it okay? Okay.
Yes.
Okay. This I think you. Okay. Maybe. Okay. We'll come to this question maybe later. Yeah. Come to this question. It's quite big. Okay. This we can solve first. A DNTP master mix is prepared by combining 40 microliter of each 20 millimolar DNTP stock, DATP, DCTP, DGTP and DTTP. 4 microliter of this DNTP master mix are added to a PCR mix and the final volume is adjusted to 50 microliter. The concentration of total DNTPs in the PCR mixes. So read the question slowly. See what is written. So it's a little bit tricky to understand, but yeah. Anyone solved? Micromolar. Okay, anyone can unmute and tell how to solve it. Let's see. Anyone wants to speak up? Anyone? Okay, let's try to do them. So what is given is each DNTP solution, yeah, is 20 millimolar, yeah, and 40 microliter is of each DNTP is combined. So total volume is 440 into 4, yeah, 160 microliter. So in this 160 microliter, each DNTP concentration we can find out. Yeah. So again C1 V1 equal to C2 V2. So C1 here is 20 and 40 and C2 have to find out, divided by volume, which is 160 total volume. What we will get is 5 millimolar, yeah, C2. Okay, so each DNTP in the mix is, when after mixing, after in the master mix is 5 millimolar. Okay, So when adding to the PCR vial, PCR mix, yeah. So at that time, PCR mix, okay. Okay, when adding to the PCR mix, 4 microliter of the mix is added. So when adding 4 microliter, okay, 4 microliter, okay, so C1 V1 equal to C2 V2 again. So C1 here is maybe 5 millimolar into, is it correct? I am doing. Yeah, so yeah, maybe we can directly do it again on the 4. We can add all the DNTP correct. So 4 microliter C2 and Y V2 is 50. So 10, so it is 0.4 millimolar, or you can take it as 400 micromolar. Yeah, so it is 1 DNTP concentration in this. But when you will have 4 into 400, because there are 4 DNTPs, you can get it as 1600 micromolar, yeah, or 1.6 millimolar. Is it clear? Anyone, any doubts? Any doubts? No? Okay, then we can then go to the next one then. Yeah. A qPCR experiment shows a CT value of 25 for sample A and 28 for sample B. Assuming 100% PCR efficiency, how many times more template was present in sample A compared to sample B? Anyone? Answers? No? Okay. So what you have to say is how much. So what you will do is usually know that, yeah, 2 to the power. So sample. So you have to, what you have to find is or sample. So essentially what you have to find is sample B and sample A, the difference here. For sample B, it was what you the cycle number was to the power 28, and then you have 25. So what we will do is 28 minus 25. What you will get is 2 to the power 3. So it is 8 times. Yeah, was present in sample A, then the sample B. So 8 times more template was present in sample A. Is it okay? So the group has suddenly gone too much silent. Uh, is there, it is understandable, yeah? Okay. So in a nested PCR, the outer primers amplify a 500 base pair region, and the inner primers amplify a 200 base pair region within this amplicon. If the efficiency of both the PCR reactions is 100%, you start with 100 copies of the template DNA. Approximately how many copies of the final product would you expect after 20 cycles of the first PCR and 15 cycles of the second PCR? Answers, please. Okay, let's solve it. So first PCR round, yeah. So first PCR round has 20 cycles. So after 20 cycle, how many number of copies? 100 into 2 to the power 20, yeah. And using this 20 cycles, after that you run the second PCR. So in this second PCR, this initial, so I told you, yeah, number is equal to number of initial into 2 to the power number of cycles, yeah. So here in this it will be the n1, which becomes the initial number of into 2 to the power 15, yeah. So here it will be 100 into 2 to the power 20 into 2 to the power 15, yeah. Is it okay? So you can simplify it more. 2 to the power 20 plus 15 into 100, you can write. Is it okay? Everyone understood? So you one of you were adding it, so it won't be added here. You will have to place it in the same formula essentially. So you can see nested PCR how it enhances the sensitivity. Yeah. Basically, on top of one exponential, you are actually running one more exponential. So it's kind of doubling. It is enhancing too many times. So that's yeah. Actually, this was the last one, and okay. So, so this is a sequence which is given. Okay. So what I want you people to do is what you can do is so this you can take it as an assignment. So you have to design one set of primers, forward and reverse, and you have to generate an amplicon such that it contains this underlined A you are seeing, yeah, that it should like the primer should take care of this underlined base. The primer should be 20 bases long. The amplicon should be must be 100 to 150 base pair in size. Yeah. The primers must have similar melting temperatures. So you have to find out how to calculate this melting temperature. Then plus minus 2 degrees acceptable. And the primer should have no homology in the last three prime bases. So that's what the complementariness should not be there. So what I want is you people take a screenshot of it. Okay, screenshot of that the slide, or you can actually get the slide directly in the next one, and then we'll because this question will have multiple answers. Yeah, everyone can have their own answers because yeah, it can have multiple answers really. So we can discuss this once we start the next class. So first thing we will discuss this thing, and then we can go ahead with the class. So is it okay? Is it clear? Can you paste the sequence? I can, but then this underline thing will go. That's why. Fourteenth question. Yeah, I'll go. Once you people just screenshot it. If everyone done, na? Yeah, everyone done. Then I can. I'll move to the one. No issue. Has everyone taken the screenshot? Yeah, the question is clear. Yeah. If anyone has any doubts in the question, you can ask now. It's kind of a simple question. Yeah, and you will understand that it has multiple answers. That's what. And multiple answers are correct actually. Here. Okay, I believe everyone has taken. So what you have to do is write the primer sequences. How you will do it is you will write it in the forward and both the reverse strands in 5 prime to 3 prime fashion, such that that's what you will get from DNA synthesis facility. Yeah? You will order it as that. And you have to also write the melting temperature, as I had mentioned here. You have to write the melting temperature also for each primer that you have designed. Is it clear? Perfect. Yeah, you are asking 14. Yeah, 14. Yeah, please. You have any doubts in this? We can explain it again. Fourteen is clear. Yeah. Yeah, anyway, if you miss it, that's the PPTs are there, so you don't need to worry. Yeah, just the assignment that is given. You can try it. Yeah, we'll see how it comes. Sure. Yeah. So that's kind of the end. So if you have any doubts, we can discuss. Otherwise, we'll meet next week.